Thank you very much for the nice introduction. Thank you to the organizers. It's a real pleasure to be here. Such a pleasure that I'm going to change my slide and put a picture from here rather than Stanford. Okay, so we're going to go back to solid tumors. And today I actually changed my talk, and I want to talk about something new towards the end. But I want to talk about early events in cancer may influence late events in cancer. And so to do so, I'm going to focus on one disease, and this disease is small cell lung cancer, or SCLC. And so you could look at the pictures and then you can read the text, but just to summarize, small cell lung cancer is the most lethal form of lung cancer. And I just want to highlight 2 points. First, it's a neuroendocrine cancer, so it's very different from adenocarcinoma or squamous cell carcinoma of the lungs. It's, again, the most lethal form of lung cancer. It kills many people. We have no therapy for it. And another aspect I want to highlight, it's a really aggressive disease, and it's highly metastatic, and you can see, obviously, an extreme example here. So maybe one reason why clinicians cannot really treat small cell lung cancer effectively is we have actually very few human samples to work on. And this is, you may not want to read that, but the reason is because patients rarely undergo surgery, because studies in the 80s and 90s, and this is one of them here, show that except in rare cases where disease can be diagnosed early, if you resect the tumor, you don't resect the tumor, the survival is the same. So there's no surgery, there's no clinical sample, so you cannot do all the studies a lot of people do, including the leukemia people or the breast cancer people. So this is really why there was a real tour de force from the group of Roman Thomas. We were fortunate to collaborate with them on this study. They were able to get 100 tumor samples from all around the world, Germany, France, the UK, the US, and Asia. And they were able to do deep sequencing on these tumors. And so you've seen this kind of clinical landscape slide before. You have all these mutations in all directions. But again, I want to highlight 2 points. The first point is that basically in 100% of cases, small cell lung cancer tumors have loss of function mutations in both the RB and the P53 tumor suppressor. So this is very different from many other cancers where initiation can be driven by one point mutation in one oncogene, for example, in pancreatic cancers. Here, you need to lose 2 tumor suppressors before cancer can start. And you can see all the other mutations. I will highlight one more, which is that in some cases, 5 to 10%, a little bit more, there are inactivating mutations in the 2 RB family members, RBL1 and RBL2, also known as P107 and P130. And actually, we've been using that in the development of mouse models for the disease. And the reason to develop mouse models is because, as I just said, small cell lung cancer is very lethal. We have no therapy. It's also really extremely difficult to study in patients because we don't have access to clinical samples. So the field was actually pioneered by a group of Anton Burns, and his lab developed the first mouse model where they deleted RB and P53, which are the 2 essential tumor suppressor genes that have to be mutated. And my lab generated what we call the TKO, or triple knockout model. So we use Krylog strategy, we delete RB and P53. We also added deletion in P130, the RB-related gene. And this gives us just faster tumor genesis and more tumors. But otherwise, the tumors are very similar to double knockout tumors and to human tumors. And so what we do is we use an adenovirus carrying the Kruger combinase, and it's like the mice, instead of smoking carcinogen with cigarettes, they just inhale the virus. So we do a hole in the trachea. The mice inhale the virus spreads to their lungs, infects the lung epithelial cells, deletes the tumor suppressors, and then the mice develop tumors that are genetically and histologically similar to human small cell lung cancer. So what we've been doing in the past few years has been to use this mouse model as well as human cell lines and human primary samples when we can get them to study various aspects of cancer initiation and progression in small cell lung cancer. Okay, so I just want to give you 2 slides because I was put in the cancer stem cell session, so I'm going to talk about cancer stem cells. But we've used this mouse model to try to identify if there are cancer stem cells in small cell lung cancer. And so we're actually going to call these tumor propagating cells, or TPCs. And so what we do, we can take our mouse model, infect the mice with Ad-Cre triple knockout mice and get tumors. Then we can get single cell suspension and transplant again into recipient mice and try to identify markers or properties of some subpopulation of cells that have the ability to transplant, self-renew, multiple passages, and form new tumors. And so just an example of the kind of data we got, we found that cells that are high for CD24 at the surface, low for CD44, and very high for EpCAM are actually the cells that can transplant and make new tumors. These cells happen to be also very high for NCAM, which is also known as CD56, which is a neuroendocrine differentiation marker. So to summarize this work in this little cartoon, what we found is in small cell lung cancer, the situation may be more similar to other cancers such as melanoma, which is nicely shown by Sean Morrison, where actually the cancer stem cells are quite frequent or numerous in the tumors. In our case, these CD24 high, EpCAM high, CD44 low cells can self-renew, they can generate non-TPCs, they represent around half of the tumors, they are really epithelial and neuroendocrine. They don't dedifferentiate, they don't lose the marker of differentiation. The more differentiated cells are actually the ones that transplant the best. And these cells are actually not quite standard cycling. And we're able to do similar work in human tumors and confirm largely these results. Okay, so what I've told you so far is that small cell lung cancer is the most lethal form of lung cancer. It's a neuroendocrine cancer, and it's really driven by these cells that have these neuroendocrine differentiation features. One aspect of the disease that we've been really interested in is metastasis, because I told you that in around 70% of cases, patients actually already have metastases at the time of first diagnosis. The beauty of the mouse model is we can actually have access to metastases. The clinical sequencing genomics that I showed you before, where 100 tumors were all primary tumors. No metastases have been studied or sequenced in patients. So for us to study metastasis, what we did was to take our triple knockout model and we crossed these mice with ROSA26 MTMG mice. So in this reporter system, the normal cell, the white type cells are red, and you can see in green the tumor cells that grow in the lungs of these mice. The beauty of the system is we can also follow how these tumors metastasize to different organs, including the liver, and you can even find small metastases that you could not see otherwise. In addition to seeing the metastases, we can use flow cytometry, and for the first time now we can isolate pure population of metastases and pure population of primary cells, and now we can compare the two populations and try to understand the molecular mechanisms of metastases. So this is work we've done in very close collaboration with a group of Monty Winslow as well as Will Greenleaf. And because this is published, I just want to show you a summary of that work. So what we found in this triple knockout mouse model of small cell lung cancer is that in some primary tumors, there were clones that would emerge, and this clones had upregulation of this transcription factor, whose gene is Nfib, and the protein is called Nf1b. So there's some advantage for these clones to grow, and these clones actually are the ones that give rise to metastases. So upregulation of Nf1b is due to amplification of the gene, and this gene actually has been involved in lung development as well as brain development. When we have more of the Nf1b transcription factor, this is associated with an opening of the chromatin, which is actually quite striking. I'll show you some data. And this opening of the chromatin is itself associated with increase in some gene expression programs, including programs that we would expect to see metastases, for example, cell adhesion and cell motility, and other programs. For example, we see more neuronal differentiation. So the tumors which are neuroendocrine become a little bit less neuroendocrine and more neuronal as they become metastases. So let me show you some data from this work. So this is again our Ad-Cre TKO model. Infect the mice, get primary tumors, the primary tumors metastasize, and the histology of the primary tumors and the mets is very similar and similar to human tumors. But you can see by immunostaining very strong upregulation of Nf1b in the mets and low or undetectable in the primary tumors. In terms of chromatin opening, we use Ad-Cre with Will Greenleaf and his student Sarah Denny. And you can see it's very striking. It's really a one-way opening of the chromatin in the metastases compared to the primary tumors. And the range of the effect is actually more than the differences between T cells and B cells, and a little bit less than what you can see between lung and liver. So it's quite a dramatic change in the chromatin level between mets and primary tumors. This opening of the chromatin drives some genetic changes, including gene expression programs associated with neuronal features. And actually, some cases in some of these metastatic cells, we see this protrusion that really look like axons. So these cells become really more neuronal as they become more metastatic. We still do not understand how that promotes metastases, though. So what we also found is that Nf1b was both required and sufficient for the metastatic program. If you take tumors which have high levels of Nf1b and you implant them under the skin of mice, some of the cells in these tumors will see metastases in the liver. If you knock down Nf1b in these primary tumors, this does not affect the growth of the tumors under the skin, but it significantly decreases the number of metastases. And actually all the mets that we see here have actually regained expression of Nf1b, so there's very strong selection to re-express Nf1b. So Nf1b is required. It's also sufficient. If you take cell lines which actually do not metastasize very well, you see very few mets in the liver, and you overexpress Nf1b, you have more mets, and it's quantified here. So Nf1b is sufficient to promote metastases. Okay, so we're quite happy with this study because it was the first evidence that we could have a molecular switch that would change primary tumor cells to a metastatic state, and hopefully in the future we would have ways to prevent that switch or block the effects on F1b. And then we did some more experiments. You should stop doing experiments at some point, but we had to continue. So this is the model I talked to you about, the CMV-Cre TKO model and F1B upregulation. We have another mouse model, and this mouse model has the same exact histology in the primary tumors and metastases. The time to metastasis is 6 to 9 months, the same as in the first model. The percent of mice with metastases is around 50 to 60% at 6 to 7 months, exactly the same. The same genetic alteration, triple knockout with the reporter, but one major difference was that there was no NF1B upregulation in the metastases compared to the other models. So low levels of NF1B in the primary cells and in the tumors. It's actually also true in double knockout mice, so it's not due only to the triple knockout system. So what is the difference between these two models? Well, there's only one difference, which is the promoter that drives Cre expression. In the second model, what we did was to use CGRP Cre instead of CMV Cre. So just 2 kb in the promoter of this driving Cre expression is different. And CGRP is a gene that is specifically expressed in neuroendocrine cells in the lungs. And remember, small cell lung cancer is a neuroendocrine cancer, so you would expect the CGRP positive cells to be the cell of origin for small cell lung cancer. And clearly it can be a cell of origin. So when we try to be more specific and control the situation and only need the tumor suppressors in neuroendocrine cells, then we got a very different phenotype, at least in terms of NF1B expression, compared to the CMV Cre. So this was really intriguing for Duane, the student in my lab and Monty Witzel's lab, and he decided to pursue this observation and compare the two models. So I can show you some quantification first. If you look at CMV-Cre TKO tumors or CGRP TKO tumors, these tumors have low expression of NF1B. In the CMV model, you see nearly all the mets have high levels of NF1B. CGRP mets, very low levels of NF1B. So really distinct models in terms of tumor progression. How does that relate to human small cell lung cancer? So this is analysis that we did with around 100 metastases by immunostaining. And you can see that around half of the human mets in the brain and lymph nodes have high levels of NF1B. And then the other 25 to 40% have low levels of NF1B. So it is possible that the two mouse models represent at least 2 different ways for the human tumors to become more metastatic in this case. So that was further support for us to study this observation a little bit more. So I don't know what you guys do, but when we don't know what to do and we don't understand, we run ChIP-seq, RNA-seq, ATAC-seq, all the seq we can do, so we did that. And then we found is although the tumors look very similar, even at the level of the primary tumors by RNA-seq, we can find around 2,000 genes that are different between the NF1B low CMV tumors, so before they become metastatic, and the CGRP tumors, which are always NF1B low. So at the molecular levels, even though the tumors look the same, they are quite different. We also run ATAC-seq on these tumors and on metastases, and we actually, very strikingly, we saw no chromatin opening in the CGRP TKO metastases compared to their primary tumor. So at the chromatin level, also the two models are very different. Then we pursued observations and we look more at metastases. So I told you earlier on that as in the CMV TKO model, as the tumors become more metastatic, they become more neuronal. And indeed, we have these GO terms, GSCA type of analyses. In the CGRP TKO model, we also find the same neuronal up signature. But when we looked at the genes that drive these neuronal signatures, there's actually very little overlap between the CMV model and the CGRP model. And when we look more carefully at the GO terms, in the CMV model, the genes are more early neuronal differentiation, they're early migrations, whereas in the CGRP model, these are more late markers of neural differentiation, synaptic signaling and GPCR. So in both cases, and we don't understand that, but there is a decrease in neuroendocrine differentiation and increase in neuronal differentiation, which accompanies this metastatic program. But actually, the programs are different in the two cases and two models. Okay, another observation we made from these experiments was when we looked at genes that would be expressed in the primary tumors and metastases, we found that in the CGRP case, you can see the tumors and the mets here, these tumors and metastases express low levels or no expression of markers of other lung epithelial lineages. So this is a marker of club cells in the bronchioles, and those are markers of alveolar type 1 and type 2 cells. So low or no expression. But in the CMV primary tumor specifically, we could have high expression of this club cell marker, CC10, pretty high expression of this surfactant gene, et cetera, et cetera. So there seems to be this multi-lineage differentiation in the primary tumors in the CMV model, which we do not see in the neuroendocrine-driven CGRP TKO model. So this was RNA. I can show you some protein data too. So again, we have 4 conditions here, CMV TKO versus CGRP TKO, primary tumors and metastases. The histology, the pathologist tell us is very similar. If you look at primary tumors and metastases, If you look at markers of neuroendocrine differentiation, for example, UCHL1, these tumors are all positive for UCHL1. If you look at metastases, NF1B is specifically upregulated in the CMV TKO metastases, not in the other model. But if you look at markers of other long differentiation lineages, you can see, for example, C-Cten expression specifically in the CMV TKO tumors and not in the other conditions. And I'm not going to show you the data, but we use lineage tracing with confetti mice and similar tricks, and we found that these cells are actually cancer cells. They're not infiltration of normal cells. They are indeed triple knockout cells that belong to these tumors. So at this point, the simplest hypothesis and the best hypothesis we have is that the two models initiate from two different cell types in the lung epithelium. And this idea is actually validated by additional observations. For example, on the left here, even though we use a titer of the Ad-Cre CGRP virus, which is 10 to 20-fold higher than the CMV-Cre, we get around 10 to 20 times fewer tumors that grow in the lungs of TKO mice. So this suggests either there's another cell of origin here which is easier to transform, or there's more of this cell of origin compared to neuroendocrine cells. We don't know that. Another example, another support for our hypothesis, if you look at the localization of the lesions and the tumors in the CMV and the CGRP models, we see tumors and lesions in both large bronchioles and smaller bronchioles in both models, but we only see lesions in terminal bronchioles at the bronchoalveolar duct junction in the CMV model and never in the CGRP model, suggesting again there might be another cell type of origin in this case. Okay, so what could be the cell type of origin? I mentioned to you that there is a population of neuroendocrine cells which can be marked by CGRP in the lung epithelium. There are also other cell types obviously in the lung epithelium. We already discussed a little bit that there are club cells, which are previously known as Clara cells, that are CC10 positive, or alveolar type 2 cells, which express surfactant protein, for example, SPC. These are more alveolar, these guys are more bronchial. And so we started to try to determine which would be the cell of origin in the CMV model. So to do so, what Duane did was to use various adenoviral Cre vectors that were generated by the group of Anton Burns. So in this cohort of mice, if you infect these mice with adeno CMV-Cre, we get these 40 to 50 tumors that grow in the lungs of the mice. I told you that with the CGRP-Cre, although we use 10 times more virus, we actually only get 1 or 2, sometimes 3 or 4 tumors. So what Duane did here was to infect the mice not only with adeno CGRP-Cre, but CGRP-Cre plus C-Cten-Cre, CGRP-Cre plus C-Cten plus SPC. And in all these two additional cases, actually did not find an increase in tumor numbers. So to us, what that means is that the cell of origin in the CMV model is not CGRP positive, not C-Cten positive, and not SPC positive. What is the identity of the cell of origin? You want to see the result on the next slide? It's not there because I don't know. I want to summarize that. So I think what I told you today is there's maybe two messages. The first one is different cell types of origin may give rise to tumors that look the same by histology, which is, I think, a first observation. The second observation, which is actually conceptually even more interesting, I think, is that the early events in cancer initiation can very strongly influence the molecular mechanisms by which tumor progress, especially in metastases. And remember, metastasis in this case happens 6 to 9 months after Ad-Cre infection. So some have as an epigenetic memory of the cell of origin that strongly dictates how the tumors will progress. So just to give you more details, the classical view of small cell lung cancer could be that it's a neuroendocrine cancer. So you start cancer in CGRP positive neuroendocrine cells. You get small cell lung cancer very easily. These tumors are neuroendocrine. As they metastasize, they're still neuroendocrine, and they are a little bit more neuronal than they used to be in the primary tumors. So this is what people would have expected would be the classical case of small cell lung cancer. What we found when we use adeno CMV-Cre is there's another cell type of origin, maybe several other cell types of origin. They're not SPC-Cten or CGRP positive. We don't know what they are, but our best guess might be a new type of stem cell in the lung, because the primary tumors we see actually express markers of various lineages in the early stages of cancer. Neuroendocrine is obviously the majority, but there are also markers of club cells and alveolar type 2 cells. However, these tumors nearly always, as they progress, they become more neuroendocrine. And then there's a switch. They amplify Nfib, become metastatic. They are more neuronal, less neuroendocrine, and they're really aggressive metastases in this case. Okay, so what does that mean clinically? I don't know. But I think one thing that could be useful in the future is that now we may have biomarkers to predict how tumors may progress. right? So if you had a patient whose primary tumor was a mixture of neuroendocrine cells and non-neuroendocrine, let's say, club cells, you might be able to predict, based on the mouse data, that this patient will metastasize with NF1B upregulation. So if we had a way to inhibit NF1B or block NF1B action, then we might be able to preventively help this patient not get lethal metastases. On the other direction, I'm not going to show you any staining, but we do actually have around 1,000 markers that are specific for this state versus that state. And so again, we could predict that this patient will progress using this other mechanism, the non-NF1B mechanism in this case. So in the future, this may be helpful. So I'm going to stop here. I really want to acknowledge my whole lab. Nadine Jachin was the postdoc who spearheaded the cancer stem cell mechanism. But this project was really led by a very talented graduate student who's now a postdoc with Jonathan Weissman at UCSF. And Duane was actually a shared student between my lab and a group of Monty Winslow. And I have to give a lot of thank you and for Monty's effort intellectually and technically. This was really a shared project, and it's been a real pleasure to collaborate with Monty. So we've had other collaborators, I think I mentioned. I'm going to stop here. I'm happy to take any questions. Thank you. And I can leave the model because it's quite complicated.
Thank you, Dr. Sage. I think maybe I'll start with my question so I get to ask it. I'm curious about the Nfib, really fascinating work in the liver mets in your one model you showed. And I noticed you showed in the human samples that you had that there was Nfib, and you mentioned brain.
Yep.
Do you think the Nfib upregulation is specific to liver? Is it enriched specifically in liver, or will it occur as
this is
a
really key question, I think. It's really, thank you for actually, I should have said that. So the tumors become more neuronal, and small cell lung cancer is known to metastasize to the brain. So there could be something there where more neural differentiation might help the tumors go to the brain. However, as far as we can tell, we see NF1B high mets in the liver, in the lymph node, in the brain. So right now we don't have any support for this idea that it could be some tissue-specific interaction, but maybe we haven't explored that enough. So it's a really good question. Yanis?
Related question. How do you think NF1B is induced? I mean, you have the attack sequencing data. Can you see transcription factor motifs in the opening?
Yeah. So in the mouse model, it's very clearly gene amplification. This region of the genome is amplified. So we can see that from the attack-seq, and we can do other methods. In humans, it's actually not amplified, but it's highly upregulated at transcriptional level. So my first hypothesis was a loop, but we did ChIP-seq for NF1B. It does not regulate each other. Then we thought MYC, because MYC is high in small cell, and we cannot find a big binding site. So the factor that upregulates NF1B, we don't know what it is. But in humans, it has to be some transcriptional program.
And I think NF1B is oncogenic in brain tumors, like neuroblastoma, isn't it?
Yeah, so it is oncogenic in some tumors. It's actually tumor suppressive in other tumors. I forgot which one, one of the pediatric cancers. So it's not going to be universal pro-metastatic factor, but it could be from the brain cancer that there are related mechanisms of action, which we haven't explored yet. It's a really good question, too. The back. Yeah, sure. I can repeat.
Assuming that the mets in the two models are clonal, is there any convergence of secondary hits detected by sequencing the mets between the two?
Yeah, so the mets are actually clonal. So like even in the CMV model, we get 50 tumors. Usually only one of those tumors forms mets, and we use confetti and all this different lineage tracing. So, and that metastasis, comes from one aspect and from one cell, as far as we can tell. Although it could, can go first to lymph node and then liver, so it can jump from different organs. In the CGRP model, it's much easier with fewer tumors, also very clearly clonal from one primary tumor that seeds. In terms of the mutations they accumulate, it's very interesting because we've sequenced with Roman Thomas the CMV TKO model, and the mutations the mouse tumors accumulate are actually the same that the human patients accumulate, that recurrent mutations. So at the mouse model, we get Notch mutations, MLL mutation, P300 mutations, very similar to the recurrent mutations in human patients. The CGRP model, we haven't sequenced there, these genomes yet. We do see loss of P10 and amplification of MYC, similar to what we see in the model, but we haven't done the point mutations. So we don't have any evidence that would be different there, too. Very early work still. Right, thank you so much.